Hello, one and all, and welcome back for more of endocrine physiology. So, what we're going to start talking about now is a little bit of hodgepodge stuff that wouldn't fit in anywhere else, but the main thing that we're going to try to discuss here is interactions that hormones have with other hormones. So, let's go ahead and dig right in. So one of the things we mentioned in the last video, and we talked about this a fair bit in the communication unit, a lot of these signal transduction pathways will involve second messenger systems that use a type of enzyme called a kinase. So I wanted to go ahead and formally go over this with you now. So a kinase is an enzyme that does one very specific thing. It takes a molecule of ATP and transfers one of those phosphate groups, one of the three of them, and transfers it over to one of the amino acids that make up a protein. So a kinase is going to take a protein that is not phosphorylated, which you can see up here, and after doing its thing, it adds the phosphate to the protein and makes it a temporary part of the protein itself. The reason why this is important is because adding or removing phosphates like this is kind of like flipping a switch on the protein's activity. A protein may be completely inactive or in an off position, as you can see here, if it doesn't have a phosphate, and when a kinase adds a phosphate, it could turn the protein on and actually give it its activity. Or the opposite is true as well. A protein may be completely active when it is not phosphorylated, and it may be shut off when a kinase adds a phosphate group. So this really is like flipping a switch. We can either turn a protein on or turn a protein off just by having a kinase add that phosphate group. Now, we can also remove phosphate groups from proteins, but a kinase does not do that. A separate enzyme called a phosphatase is going to do that. So if you can just remember that kinases add phosphates to proteins and phosphatases remove them, then you should be all squared away. So when considering how active a protein is, it's all going to be determined by a balance between kinases and phosphatases. And generally speaking, a lot of the signal transduction pathways that are the result of hormones that we'll be talking about will activate kinases like protein kinase A, which we have discussed before. Yeah, so protein kinase A is a part of a lot of G-protein coupled receptor pathways that are going to rapidly phosphorylate proteins inside the cell to give us some kind of change in cell physiology, which, as part of a whole tissue, those cells which have new function should give us our overall physiological response. And we definitely don't want to forget about a topic that we discussed in the communication unit. Not only do you need signal transduction, so this picture shows this concept, and this picture was also in the communication slides. Not only do we need signal transduction, so not only do we need to get that message from the extracellular side of the membrane to the intracellular side, but we need to amplify that signal as well. And in a lot of our hormonal signal transduction pathways, the amplifier here is going to be an enzyme called adenylyl cyclase. So that is going to be an enzyme that rapidly produces the second messenger, cyclic AMP. And you'll also recall in a separate G-protein coupled receptor pathway that we looked at, a different type of amplifier enzyme called phospholipase C produced two different types of second messengers called IP3 and DAG. DAG in particular being interesting because it stimulated the endoplasmic reticulum to release all of its calcium, which also acts as a second messenger. So in a number of different hormonal pathways, we may see a few different types of signal transduction pathways occur, but just be aware that in the case of something like what we're seeing here, the end result is probably going to be in some way or another activating a kinase and changing the function of intracellular proteins. Another thing that we discussed a little bit in the communication unit, and it definitely is relevant for our discussion on hormones, you'll remember that we said that the strength of a ligand-receptor interaction is going to be dictated by the number of receptors that get activated by a given ligand, and this is certainly true for hormones. So you'll recall that we had a discussion on upregulation and downregulation, basically the way that the cell kind of modulates its sensitivity to a particular response. So there are kind of two extremes to this. So one extreme, a cell may have no receptors, right? So the idea was the fewer receptors a cell has, the less sensitive it's going to be to a particular response. So obviously if a cell has 0 receptors, it's not going to be responsive at all. And this is what we call a non-target cell, meaning a cell that cannot possibly respond to a given hormone. And like I said, as we've discussed, a cell can kind of toy around with how many receptors it actually expresses on its membrane, and the more receptors that there are, the stronger a hormonal response can be. The fewer receptors there are, the weaker the response is going to be. And if we get that number down as low as 0 receptors, like we said, you're not going to get any response at all. Okay, so now let's go ahead and start talking about these hormonal interactions. So here is the deal. We are going to talk about hormones in the rest of this chapter, mostly on a one-by-one basis. So we'll spend some time talking about insulin, we'll spend some time talking about thyroid hormone, we'll spend some time talking about estrogen. So we're going to talk about all these hormones on kind of a one-by-one basis. But the important thing to understand is that the bloodstream does not contain one hormone at a time. The bloodstream at any given time is going to contain dozens of different hormones. And we would be remiss or naive to think that each one of those hormones does not have any effect on the other hormones that are also present in the blood at the same time. So one hormone can definitely have a strong effect on how another hormone does its job. And that's what we're going to try to understand here. So hormonal interactions come in 3 different varieties. The first is called synergism. So if we've got a set of synergistic hormones, we've got a set of different hormones that generally all do the same sort of thing, but when they're all present together, they can all produce a stronger response than each one of them would have done individually. So the best example of this is 3 hormones, glucagon, epinephrine, and cortisol, and we're going to talk about each of them as we kind of go along through the chapter. All you need to understand this particular concept, though, is that each one of these 3 hormones, they all have the overall effect of increasing the amount of glucose in your blood. So they raise your blood glucose. All 3 of them individually will do that. You can see that's the case because this graph that you're looking at here, on the y-axis, we are tracking the levels of glucose in the blood in milligrams per deciliter. And then on the x-axis, we can see time in hours after a particular hormone was administered. So if you look at each of the individual graphs, you can see on the bottom here in the purple graph, cortisol all by itself, assuming it is the only hormone in the bloodstream, very modestly raises the blood glucose. So it's not that strong of an effect there. Epinephrine in the green graph has a slightly stronger effect, but it's still nothing too terribly impressive. But it is still raising the overall blood glucose levels. Same deal with glucagon. It raises the blood glucose very slightly. So all 3 of these hormones are producing the same effect, an increase in blood glucose. But look at what happens when we allow these hormones to be in the bloodstream at the same time and work together. If we have both glucagon and epinephrine, then we get a much more impressive increase in blood glucose. And then look at the dark blue graph up here. We have all 3 of these hormones acting together at the same time. We get a super impressive increase in blood glucose, certainly much more impressive than we got with any one of those hormones acting all by themselves. So that is synergism. The other 2 types of hormonal interactions are called permissiveness and antagonism. So permissiveness is going to sound an awful lot like synergism, but it's not quite the same. So permissive hormones will strengthen the effect of one another, but the difference is that with synergism, each one of those hormones all by themselves, they were still able to do their job, they just didn't do their job very well. With permissiveness, you have to have these hormones together, or individually they're not going to do anything for you. So, for example, some of the reproductive hormones that we're going to talk about, like testosterone and estrogen, they can't really do their job at all unless the thyroid hormones are also present. So both of those things have to be present in order for things like testosterone and estrogen to do their job at all. And then finally, antagonistic hormones. The best example of this is insulin and glucagon, which we're going to illustrate at the bottom of the slide here. So antagonistic hormones, similar to synergistic hormones, are both going to have control over the same parameter. In the case of insulin and glucagon, that parameter is blood glucose. But antagonistic hormones are going to have control over the same parameter, but produce changes in opposite directions. So the idea here is that insulin, when it binds to an effector like the cells of the liver, it is going to lower your blood glucose. Glucagon, on the other hand, when it binds to its effector, it's going to have the opposite effect. It's going to raise your blood glucose. So the typical thing that you see with antagonistic hormones is, number one, they have control over the exact same parameter. So in the case of insulin and glucagon, that's blood glucose. Number two, whatever changes they produce, they are going to be opposite for one another. Insulin lowers your blood glucose, glucagon raises it. And then finally, the consequence of the two things that we led into with this, you're not likely to find antagonistic hormones in the bloodstream at the same time, because that would be kind of a zero-sum game. If insulin and glucagon are both in the blood at the same time, insulin's trying to lower your blood glucose at the same time that glucose is trying to raise it. So you're not really going to accomplish much if that is the case. Certainly, that's not going to be useful for us for the sake of homeostasis. All right, so that is going to do it for this particular video. So we are just about ready to start talking about individual endocrine glands and hormones in earnest. I think we are just about ready for that. So be ready, come back next time, and we will start talking about how negative feedback works in the endocrine system, and we are going to introduce the two biggest players in the whole endocrine system, the hypothalamus and the pituitary. So I will go ahead and sign off for now. See you next time.